So now, after collecting the water sample, we are going to transfer this into our MIMS vials. As you can see, we have 3 different vials for our 3 replicates, just to be safe. And so in each vial, we are going to take 40 mils and transfer it into the vial. And so basically we're at 140, we're going to go down to 100. From 100, we'll go to 60, and from 60, we'll go to 20. And so at the end, we should have about 20 mils left in the syringe. And so to start, again, we have our stopcock. It's closed here because we don't want anything coming in, anything going out of that way. But we have the arrow pointed up to our syringe and the arrow pointed out of our tubing. And this is great because we are going to uncap these. And then once uncapped, we take this tubing and we place it, and it's touching the bottom of our vial. And from there, we are going to push our first 20. So we're at 140, and we're just going to push 20 milliliters out of the syringe of water. As you see, it's going to overflow, but that's okay. Okay, we're at 120, and from there, we're going to grab our tubing, and we're going to keep pushing 20 more. But we're going to slowly pull it out. And we're going to stop it, so no drip. We're going to put it again, the tubing at the bottom of the second vial. So now we're at 100 milliliters. We're going to go down to 80. And again, it's going to overflow, but that's okay. It's kind of cleansing the vial. Okay, now we are at 80. I'm going to grab this tubing, slowly pull it out, and push out another 20 mils. And I'm going to stop it once I get to 60, no drip. Our final replicate, put it at the bottom of the vial again. Now we are 60 milliliters, push down to 40. I'm at 40, grab the tubing, again slowly push out the water. And we will get to 20 mils here shortly. And then stop pushing once you get to 20. And so that is a water transfer, but one note we want to make is that when you look closely at these, the meniscus, it has the bump or the bubble over it. And so that's good because that means we're trying to avoid any bubbles, anything that could contaminate our sample. And from there, we will do our preservation, which is using zinc chloride to preserve our samples and then place it into our water bath. Well, not our water bath, but our water thing. And then we will put it in a cooler until we can get it to the nearest lab and the nearest walk-in fridge. But yeah, that is a transfer.